Hey guys, welcome to another video where we'll be talking about a very special enzyme called ubiquitin. Ubiquitin is among one of the, I would say, top three enzymes of mine that I love. Actually, never mind, forget that. I have so many, it's almost ridiculous. But ubiquitin is a very special protein just because of its functionality. Now, before I get into ubiquitin and some of its special powers that it does in our body and, a lot of living things, I want to talk about what a protein is. A protein can be used for structural, or it can even be used as an enzyme, which speeds up the rate of reactions. If you guys want to learn more about proteins, go ahead and check one of the videos that I made before, and it will give you a more in-depth look into what a protein is. So just a quick recap, a protein is a molecule that is made up of smaller amino acids. And when these amino acids come together, they form these long polypeptide chains. And eventually what can happen is there's side chain reactions within these polypeptides, and they start to fold, and they make very cool 3D structures like you see here. Or if I zoom in right down here, where you can see we have a whole bunch of helices forming, and then there's some folding, and eventually if we skip over, it becomes a functional protein. So that's kind of what proteins are. Just something real quick. Again, I do have a video on this. I just wanted to give you guys an introduction of what a protein is. Again, it's made up of amino acids that form these long polypeptide chains, and then they become functional proteins, which can be used in our body and in other living species. So one thing I like to talk about and rave about with ubiquitin is, these proteins have to have a certain look. Again, if they don't look a certain way, they can't be used. Ubiquitin is the fashion police. If you see down here, there's plenty of different proteins, some that are responsible for transcribing DNA, gene regulation, again, structural proteins. There's protein in your hair, there's protein in your fingernails. And again, if they do not look a certain way, ubiquitin is going to come in and say, nope, you guys cannot be used. You guys are not in the right shape. You guys, don't have that special look. You guys aren't folded correctly. So ubiquitin can handle all of that. With proteins, it's all about getting recognized as well. If a protein is not recognizable, it cannot be used. So let's go over to the next slide so you guys can get a better idea of this. So I have up here, it says, what is ubiquitin? So here's a bit more about ubiquitin. It's a small regulatory protein responsible for the production and destruction of proteins. Now, when I say regulatory protein, it means it regulates. It really is like the fashion police. It regulates protein production and destruction. So it is able to help. Notice how I say help. And it'll make sense why I say this later. It helps in the production and destruction of proteins. So it is responsible. It isn't 100% responsible, but there is some responsibility there. And again, I'll get there later. It is made up of 76 amino acids. Again, proteins are made up of amino acids. So when you have these amino acids linked together, you have 76 of the amino acids. It could be leucine, glycine, isoleucine, valine. These are all different amino acids that, form together. And then eventually, once all of them come together, they form this not structural protein, this like 3D protein that can be used, correct? So next we have ubiquitin proteasome system, which will be in the next slide. And really what I want to dive in here, I want to zoom in right here because this is the very, very important part. And this is where I said that ubiquitin is a helper. It's help. It's a part of the responsibility. It tags faulty proteins. So ubiquitin is not necessarily destroying proteins. What it does is it tags these proteins that are non-fashionable, right? faulty proteins, meaning they are not good. They may not be folded correctly, they're not functional. So what ubiquitin does is tag them, and it's like poking at someone. Hey, this guy, he's not fashionable, he's not cool, he doesn't look good. And that's when another enzyme comes in, and it will then degrade that non-fashionable protein. So again, let's go back because I really want to stick this into everyone's head. Ubiquitin is the fashion police. Proteins must be in a certain shape to function. Any protein that is not will be considered faulty or non-fashionable. They have to meet that fashion requirement so they can get recognized and be used. But if they're not recognizable, here comes proteasome to come and degrade that. So if we go into our third slide, ubiquitin proteasome system, and I have some very cool schematics that may look a little confusing, but we're going to go over it. I just want to throw them here so they're looked at. So this is called literally the UPS. So that's why I put a UPS truck, just to just add a little pizzazz in the slide, right? So the ubiquitin proteasome system. So this is the system here where we have a protein. And we can say that this protein is not fashionable. It just looks ugly. It doesn't have the structure. It doesn't have the folds. It completely failed the red carpet. So what we have are three enzymes called the E1, the E2, and the E3. Wow, how easy, right? Just 1, 2, 3, E1, E2, E3. And each of these have a certain responsibility within the system to get ubiquitin to tag to that target protein. So this non-fashionable faulty protein is considered the target protein. The target protein is the protein that's going to get degraded. It's going to get destroyed because it's not fashionable. So what happens is, after this E1, E2, and E3 bind these ubiquitin to the target protein, now it's recognized by a proteasome, right? Which is an enzyme that degrades faulty proteins. So you see there, that's where I don't want to confuse you guys. Ubiquitin is a regulator. It's responsible for destroying proteins. It's its responsibility, but it doesn't directly destroy. What it does is it helps within a system of protein degradation where it tags, you see, it tags a whole bunch of ubiquitin. It's like, hey, this guy's no good, this guy's no good, like sounding the alarm, tagging. So that proteasome says, okay, ubiquitin, okay, we see there's a faulty protein. You called it out, let's go get them. And then what will happen is that faulty protein will then be degraded into peptide bonds or other amino acids that can be used in another process. So it's recycled. Wow, isn't the body smart? So here's another schematic. I really prefer this upper one to the left over this one, but it's the same thing where we have our E1, E2, E3 in a series of reactions and binding. And then we have the ubiquitinated protein with a ubiquitin. And then our proteasome saying, haha, we found you. Let's go ahead and degrade that non-fashionable, terrible red carpet dress person wearing person. And let's create these peptides. All right. So now let's go into more specific. I wasn't going to stop there for you guys. I want to keep this very simple for everyone, not too in depth, but let's go into the E1, E2, and E3. So ubiquitination is what it's called. It's in 3 steps. Simply activate, transfer, ship, ATS. Just remember that. E1 is responsible for activating the ubiquitin. E2 is the transfer, the conjugating enzyme. It's responsible for the transfer. E1 is transferred over to E2. E3 is the ligase step or the ship. Takes it over to the proteasome recognizes the complex. So ligase, again, it means just binding two molecules together. You guys can look at this schematic here if it helps too. However, I have a better one in the next slide, but I just want to get you guys used to looking at these schematics and different ways of drawings and interpretations, and just different schematics that you guys can pause the video and look at. All right, so let's get into the E1. This is one of my favorites. I actually used this photo in my first book that I wrote. And this seems to me the best schematic, just in my opinion. So the E1, ubiquitin is attached to the enzyme by thiester bond linkage, powered by the hydrolysis of ATP. Just real quick, I want to mention energetics, because energetics is always important. Proteins and enzymes must be activated first. Many of them do, because how these molecular machines and enzymes and proteins and all these different molecules usually work is they're not going to do work if they don't have to. They only do it when they need to. And most of the time it's activated by ATP. So it lowers the activation energy and allows the reaction to go through by being activated. So if we go over and zoom in, blow up this image here, we have the E1, we have ubiquitin, and through the power of ATP, the most important currency in all of biochemistry, we're able to bind the E1 and ubiquitin together. So the E1 is the ubiquitin enzyme, E1, activating enzyme, and it's able to bind the ubiquitin. This E1 transfers the ubiquitin over to the E2. E2, the activated ubiquitin is transferred to enzyme E2 by attaching to its cysteine. So as you see, again, we had the E1 bind to the ubiquitin. That E1 transferred the ubiquitin to E2. And if we go down into the middle, we're skipping over a bit to the E3. It sees the target protein, the unfashionable one, the one that wore that ugly suit, the dress, non-fashionable, and then that binds. So you can see where we have something going on here, and then something going on here, and then this is our midpoint. This is the most important point. So our faulty, non-fashionable protein finally meets our E2 and E3 that bind together with that ubiquitin there. So this is very important. I call this I don't know if there's a term for it in biochemistry, but to help you guys, I call it the E2 to E3 target complex. Oops. Because that's what is formed. You have the E2, the E3 target protein, and then the ubiquitin. Now, if we continue the E3, I kind of skipped ahead. The E3 shuttles the ubiquitin to a target protein. And then we have the proteasome that's able to recognize this. So, like I said here, we have the E2, E3 target complex. And now this is the most important step. This process is repeated. So this whole process here is repeated multiple times. I believe the minimum is about 5 times or so until that proteasome is able to recognize, because again, this proteasome doesn't know that there's a faulty protein out there. That's what ubiquitin is doing. It's tagging that non-fashionable person. So when this happens, what we have is repeated rounds, and this is called polyubiquitination. So poly, kind of like polypeptide, polyubiquitin. So we have all of these ubiquitin tags. Like I'm talking about, we have so many critics that are piled up on this fashionable person. Like, hey, you're not good. And the proteasome then comes over and is able to degrade that faulty protein through the power of ATP, our currency. And then we have this degraded material, which can be amino acids, polypeptides, used for later processes. And you see here, I did write minimum of 5 to get recognized by proteasome. And that's really it, guys. That's what ubiquitin does. One of my favorite enzymes, big time, proteins, just because its ability to tag these faulty proteins in life, get rid of them, and have those the faulty protein be brought down into amino acids that could be used for something else. So that's how ubiquitin works. Again, the main idea is it tags proteins, and it works in conjunction with proteasomes, and it is a regulator. So it doesn't necessarily do the destruction, but it helps or is also responsible in the overall system of protein degradation. So I'm just going to slide back. If you guys want to, screenshot, read over it again, you guys can always play this back. So that's just a quick idea of what ubiquitin is and the ubiquitin proteasome system. And that's pretty much it. If you guys have any questions, please comment down below, like the video, share it with a friend. I really do enjoy making these videos. Hope to become a professor one these days, hopefully, but I do it through these videos. So again, leave a like, comment, subscribe, do whatever you guys have to do, and I'll see you guys next video. Peace.